Have you ever imagined a world without enzymes? These two pictures shows the difference. The left one is a world that we have enzymes, in which our biological reactions that happens in our body happens very quickly. But if there's a world that we don't have enzymes, just like the right picture, we will have the traffic jam reactions happens in our body. All the reactions will be happen slowly and we can't manage to happen them all at one time, which means slower metabolisms, limited cellular functions, and inefficient digestions. There's also finds saying that without enzymes, biological reaction essential to life takes 2.3 billion years, which means the world is possibly no life in that world. Enzymes are life catalysts. They can decrease activation energy of the catalyst reactions. Therefore, we can happen the reaction more quickly. There is this substrate specificity characteristic of enzymes. We can see from that different enzymes will have different active sites. Therefore, there's a different substrate can fit in that site, and then they can happen reactions, and we can get products. The regulations of enzyme activity is very important. The cofactors, for example, they are the non-protein enzyme helpers to help with enzyme activity, and also enzyme inhibitors, which will inhibit enzyme activity, and allosteric regulation of enzymes, which will inhibit or stimulate the reaction. Feedback inhibition is very important as it will use the product of the enzyme activity to regulate enzyme activity. So, what's the effects of environmental factors on enzyme activity? Now, let's move on to the lab experiment, which is measuring lactase enzymatic activity and about the impact of the reaction time on the reaction. Lactase is an important enzyme that will happens used to in the lactose hydrolysis. By the hydrolysis of the lactose, we will get glucose and galactose. Now, how the lactase activity is measured? A UV visible spectrophotometer is used in this process. It consists of a light source emitting light across the UV and visible ranges. Also, we will measure the detector. The detector will measure light intensity before and after the sample. So then we will calculate the absorbance based on the discrepancy in light intensity, which is show this IO and IT. This absorbance data is then analyzed to identify substances and determine their concentrations. This is a very important mechanism. So in this lactate activity experiment, we will use OMPG because the product OMP has this slightly yellow color. So we can use this to measure the light absorbance. This is how this experiment is designed. Firstly, we can see there's control groups, which is aimed to tell us that we need to have substrate and enzyme to happen this enzyme activity reactions. And then there's four groups to measure different reaction times: 30 seconds, 60 seconds, and 90 seconds, and also 120 seconds. Now, we got the impact of reaction time. From this graph, we can see that as the reaction time gradually increases, the enzymatic reaction product will also gradually increase. However, in the real world, the product will not always increase because the substrates and enzymes, they have the limited number of them. So, to some point, the product will not increase anymore. There are also other factors. The temperature. Most of enzymes will have their optimal temperature in the reaction. Different enzymes may have different optimal temperature. If the temperature is lower than optimal temperature, as the temperature increases, the reaction rate will increase and the enzymatic activity will also increase. However, if the temperature increases too much, the enzyme may have the denaturation, which means the enzymatic activity will gradually decrease. So it's not good if the temperature is too low or too high. pH is another important factor. Different enzymes have different optimum pH as well. For the pepsin, they have the pH at 2, and urase has 7. They have different rate of reaction at different pH. If the pH is lower than optimum pH, the reaction rate will generally lower than optimal pH time. And if the pH is too high, the rate of reaction will also decrease. Now, let's move on to the real-world connection. The enzyme laundry detergent is one of the very important application of this enzymatic activity. For example, the proteins that added into this detergent can move protein-based stains, and amylase, they can move starch-based stains, and lipids, they can move oil, fat, and fat stains. However, one thing we need to pay attention to is that these enzymes can also break down proteins that present in delicate fabrics, which is we need to pay attention to when we use those laundry detergent that has proteins, because they may also lead to damage or weakening of the material. So we need to pay attention to those proteins, enzymes impact on the clothes as well. Meanwhile, we have heard that we can use warm water to wash this clothes by using this laundry detergent, because it's also the temperature that's around our own body temperature. The active enzyme in laundry detergent work best at the temperature because they naturally occur in our digestive systems. So using warm water can often provide a suitable temperature for them to work. Now let's dive on to the closer to this detergent. We can see that it added enzymatic blend, which is stain remover, and also sodium hydroxide, which is mineral-based pH adjuster. We have known that the pH will impact the enzymatic activity. Therefore, the pH of the laundry detergent is also very important, which is the reason why we will add sodium hydroxide. So different factors will impact enzymatic activity, and when we apply it to the real world, for example, this enzyme laundry detergent, we also need to pay attention to them. So this is all what I want to share related to the enzymatic reactive and those factors that will impact them. I want to thank Professor Lee, who guided me to learn a lot related to the biology in this session, and also my family. They support me in this session and help me when I have some difficulties, and also my secretariat group members. Thank you for your fantastic ideas and your help for different secretariat questions. And also others who help, and my friends and other people who helped me in this class. This is my reference. Thanks for listening.